आइए हम बात करेंगे सांगर्स मेथड ऑफ डीएनए सीक्वेंसिंग की। जैसा कि हमने पहले बात की, इस मेथड में हमें डीएनए स्ट्रैंड की सिंथेसिस चाहिए और उस सिंथेसिस के लिए वी हैव टू प्रिपेयर अ टेम्पलेट। सिंगल स्ट्रैंडेड डीएनए needs to be prepared before we can proceed further towards the method of sequencing. Theory or practical, these are two different things. In theory, it seems DNA sequencing is very easy process and it should work well. Jabki practically the scenario is totally opposite. This method needs number of checkpoints, precautions to get the successful results. Otherwise, the results practically are very disappointing because of number of errors or problems which come up after the sequencing reaction is complete. हमें जब आइसोलेशन करनी है किसी भी डीएनए की, तो आइसोलेशन के बाद एक स्टेप आता है प्यूरिफिकेशन का. So far we don't have any automatic system available to isolate and to purify the large number of plasmids or large quantity of DNA at the same time automatically. So we have to rely upon the conventional methods of isolation and purification, which are like midi prep, maxi prep or mini prep. And when we choose these methods, then the efficiency is compromised because these methods involve number of chemical steps, which we have to perform using different chemicals. So ultimately the DNA is exposed to number of hazards, number of threats to get broken down or to get any kind of other structural changes. Sequencing में जब हम hurdles की बात करते हैं, number of steps are there or the points where the hurdles can be observed. जो common complaints हैं, they are like blank images. When you complete the reaction, complete the sequencing, and you end up with a blank image or with weak bending pattern that your amplified or your DNA is very weakly observed in the form of the band, short read lengths, premature termination of the sequencing part of the DNA, or strong stop bands in the well of the gel during electrophoresis. So these are all common complaints which can be recovered, or which can be these complaints can be solved by taking precautions, by taking extra care during the choice of the material, choice of the enzyme, and choice of the other methods which you have to perform. अगर हम polymerase की बात करें, we start with basic polymerase and then we end up with the different modified polymerases. Now the improved polymerases have increased the chances of good sequencing. If we use the improved polymerases and we go for the cycle sequencing, the quality of DNA sequencing has greatly improved. Then the key of success is the quality of the template. If we start with an impure sample, success rate is low. So we have to be careful that the DNA sample should not be contaminated. There should not be any small DNA fragments which eventually can act as the primers and can cause non-specific extension. And the starting material, dideoxy nucleotide triphosphate or DDNTP sequencing, Sanger's method has to be performed with double-stranded DNA, because when we go for the denaturation, then we come across with the template that is used for the synthesis of new strand, which is later on used for sequencing.